Dr. Sonal Jain from Department of Anthropology, University of Delhi. In this series of forensic science lecture, today we are going to discuss about various biological fluids and stains. When we talk about the various biological fluids which can be found at the crime scene, it could be blood, saliva, semen, urine, feces, etc. But the most common encountered biological fluid is blood. Blood, when we found at a crime scene, whenever any forensic scientist collect the evidence blood and the blood sample is being sent to the laboratory, the major questions which are being asked is the substance blood. If it is blood, whether this blood belongs to human or any animal, and if the blood belongs to human, whether it could be associated with a particular individual. So, in order to answer these questions, whether the substance is blood or not, if the blood, it is blood, then it is human or not, or if it belongs to a particular individual. So, we have to conduct various series of tests. The first test we have to confirm whether the substance is blood or not is a preliminary test, is known as Kastle-Meyer color test. It is also known as phenolphthalein test. In this test, we mix phenolphthalein and hydrogen peroxide, and the suspected blood stain is mixed with that. If a positive reaction is there, it will be indicated by a deep pink color. As I have mentioned, this is a preliminary test. So, the Kastle-Meyer color test will tell you that it is blood, but the results can be false positive. To confirm that, we have to go for certain confirmatory tests. These confirmatory tests are Takayama or Tickman test, which can be confirmed using microscopic analysis. We have certain kinds of crystals in those tests, and then we can confirm that this substance is blood. The presumptive test or preliminary test of blood is not definitive. The other testing must be done to determine if the substance is truly blood or not. Substances other than blood which may produce the pink color change, it is called a false positive result test. Apart from other preliminary tests, we have hemostasis and benzidine color test. Mostly in forensic sciences lab, if we talk about the preliminary test of phenolphthalein and benzidine, phenolphthalein is much more preferred because benzidine is carcinogenic in nature. Another preliminary test which we follow is hemostasis test. In this test, it is used by various field investigators. That means this test can be performed at the time of the crime scene only. Various field investigators can perform this test on the crime scene, where they will take a dipstick which is being moistened with the distilled water and is placed in contact with the suspected blood sample. If we have a green color change, which will indicate a positive result. So, just to repeat again that this is also a preliminary test, and there might be certain other substances which can give a green color test with hemostasis. Another major example to identify blood major test is luminol test. This is a presumptive test also which is used for blood, and it produces light when it reacts with blood rather than color. Mostly luminol test we use where large areas can be quickly screened for blood by spraying the luminol reagent on a suspected item or area. The most positive point of the luminol test is that if the area is being washed with water, still the luminol test will give a positive reflection or a positive, we have a positive color. So, in a case where supposedly a crime has been occurred and the criminal has tried to wipe out the crime scene, wipe out the blood sample using water or maybe little bit of any washing reagent. But there are certain creases, or if the blood remains are still there on the floor, and if we spray luminol reagent, then that area can be viewed for the emission of light, and we call it that process as luminescence. This test is extremely sensitive and it is capable of detecting blood stains that have been diluted up to 3 lakh times. So, any washed area can be, if we spray the luminol reagent, it will show the luminescence. This test was also used in the case of Arushi murder case, where they have washed the area, but when the luminol was sprayed, the luminescence was observed, which clearly said that there was blood at a point of time which was being washed off. and it does not interfere with the subsequent DNA testing. So, whenever if there is a sample and if we have sprayed the luminol reagent, the same sample can be used for the further DNA analysis. Now, coming to the second step, after we confirm that the substance, the red looking substance which is found at the crime scene is blood only, then we come to the next step, which is to find out whether this blood belongs to human or animals. Now, why this question arises? Because sometimes criminal, they try to fake up a crime scene. When they try to fake up a crime scene, they might use blood which are easily available. Those easily available blood belongs to chicken, pork, which can be easily taken from the slaughterhouses. So, those blood samples can be used to fake up a crime scene. That is why we have to identify that this blood actually belongs to human or this blood belongs to any other animal. For that, we have to go for the antigen antibody reactions. These tests are known as precipitin test. So, how to get the antisera? If we inject an animal with human blood, the animal will create antibodies that can then be recovered and used to test for various human antigens, including human blood antigens. So, similarly, different antiseras of different animals can be, say, in the same way they can be recovered for the testing of precipitin test. So, basically in this we use the antigen antibody reactions. We take the human antisera and the unknown blood samples in a petri dish, and if there is a precipitate, then we will get to know that this blood sample belongs to human, and if there is no precipitate, then we say that this blood samples belong to some other animal. Now, after confirming that this sample we did not observe any precipitate. So, that means this sample does not belong to any human. But now the next question is that which blood sample is being used. So, to confirm that, we have to take the antiseras of various animals and with whom the precipitation is being formed, then we can say that this particular blood belongs to this particular animal. So, after confirming using the antigen antibody test, or we can say the precipitin test, the next and major question which arises that whether this blood can be associated to a particular individual. So, the particular individual, the most common answer which we think in our mind is through the DNA profiling. Yes, that is the correct answer, but before that also we can conduct a series of tests which are far more cheaper than the DNA analysis. For example, we can go for the abiotypes, enzyme and other factors. So, until the mid 1990s, the characterization of blood stains, which includes the determination of abiotypes or enzyme markers such as phosphoglucomutase and various other factors. We all know that there are different types of blood groups which exist in humans, A, B and O and the combinations of that. Maybe the person could be with the Rh factor A positive, B positive, AB positive, O positive, O negative. But how can we say that this blood belongs to a particular individual? Maybe if we have any suspects, supposedly we have 10 number of suspects and they have different blood groups, and the blood which is found at the crime scene, it matches with one of the person only, then that makes our life easier. But if that blood groups belongs to 3 to 4 suspects, then we have to go for the further more reactions. For example, ABO is one reaction, and if we add particular enzyme test to that, that will increase the chances to further narrow down the suspects. These tests, they add statistical information through which we can reduce the number of possible resources of the blood stain. As we know, the antigens and enzymes, they occur independently of one another. The probability of a dried blood stain having a particular combination of these factors is determined by the product of their distribution in the population. For example, we have talked about the enzyme phosphoglucomutase PGM, and if we know that in a particular population that particular enzyme occurs in a specific factor or distributed in a specific factor, then the probability of combination of ABO blood grouping as well as the PGM enzyme analysis will give us a chance where we can associate with a particular individual. For example, if we say that the blood stain which is found is of type A and it corresponds to the 42% of the population. So, now you will say that that person belongs to anyone from the 42% population. But again, if we put another test to that where we use PGM1 and which corresponds to 58% of the population, but the combined effect of these two factors using the product rule 42% into 58% will give us the result as 24%. That means the probability of that person present is now in among the 24% of the populations. So, before going for the expensive DNA techniques or DNA analysis, if we combine these enzyme tests with the ABO test, then we can have or we can narrow down the list of our suspect or we can associate it with a single person. But to be very sure, we can further go for the DNA analysis. So, this DNA analysis has been most prominently used from the 1990s and it has replaced the forensics reliance on the routine characterization of blood stains to a particular individual using blood factors. So, as soon as this DNA analysis came into picture, from then we have started using this in the forensic routine caseworks. These DNA analysis gives us very accurate individualization results. But what is the negative? The only negative with DNA analysis is that the twins which are born from the same embryo, they have the same DNA. Apart from that, we can clearly say whether this DNA belongs to a particular person or not. So, to understand that, when we say that we have to extract the DNA from the blood, we must understand what is the composition of the blood. So, blood refers to the complex mixture of cells, enzymes, proteins and inorganic substances. We have a certain liquid portion which is mostly we call it plasma or serum. Serum is basically plasma minus fibrinogen, that is the clotting factors. So, the liquid portion is mainly water which accounts to 55% of the blood content. Then what is the rest 45%? The rest 45% is the solid portion which is made up of RBCs, WBCs and the platelets. Our WBCs are further divided into granulocytes and agranulocytes, where we have lymphocytes, our B cells and T cells for our immunity, and agranulocytes we have eosinophils, basophils and monophils. So, where the DNA is present? DNA is present in the white blood cells, the nucleus of the white blood cells. It is not present in the red blood cells. Why? Because DNA is present in the nucleus and RBCs does not have the nucleus. They are anucleated cells. So, for the extraction of DNA from the blood, firstly, we have to remove other substances to go and reach towards the WBC's nucleus to extract the DNA from there. The DNA protocol uses the various lysis buffer, where we have to remove the RBCs first, then we have to lysis the wall of WBCs and enter there and then reach towards the nucleus, removing all other major organelle bodies, other proteins, precipitate the DNA out of there, and that DNA then further can be used for the investigations. Before going for DNA, as we know DNA we have to make a higher copy number, we have to go for the polymerase chain reaction, and after conducting PCR then we can go for the further DNA analysis. Now, the gold standard which is being used in forensic science for the DNA analysis is the STR markers, which is known as the short tandem repeat markers. These short tandem repeat markers are there where we can identify whether this blood belongs to that particular individual or not. So, with the nature of blood, we say the antigens, they reside on the RBC surface and they impart blood type characteristics to the cell. This stimulates the body to produce antibodies against it. More than 15 blood antigen systems have been identified, which includes the ABO system, RH systems, and we use this system forensically to identify an individual. Whereas the antibodies, they destroy or inactivate a specific antigen, they are found in the blood serum. For every antigen, there is a corresponding antibody. These antibodies, they react with a specific antigen, and when this reaction occurs, we call it as agglutination reaction or clumping reaction. This forms the basis for the species identification, that a particular antibody will only react with a particular antigen, and if there is a reaction, there will be a precipitate, or there will be an agglutination, or there will be clumping of that. So, after we have extracted the DNA from the blood, we call this technique as DNA profiling. In previous times, we used to use VNTRs, which are known as variable number of tender repeats, but nowadays we have mostly shifted to STRs, and the coming research is on the SNPs, single nucleotide polymorphisms. So, this DNA profiling has major applications. Mostly these applications come for the paternity testing, maternity testing, YSTR analysis, or the cases where have to track the lineage, where there are certain chances we have to identify whether they are their brothers, sisters, half brother, there are cases for the lineage where there are problems. The cases come about the wills, that this person belongs to this grandchild of the particular grandfather or not, or the maternal grandfather. So, in all these cases, this DNA profiling we use for that. Mostly in criminal context, such tests can be required which follow the sexual assaults. For example, to identify the father of a child which conceived as a result of an alleged assault. So, if a female deliver a child after a sexual assault, then through the DNA of that child, we can identify who the father was. And similarly, this test can be used to confirm whether the two individuals are genetically related or not. These tests are thus useful also in the cases of abandoned children, child swapping or infanticide. So, the result of the DNA test is admissible in court. In civil litigation, it involves claims by an estranged partner for financial support and maintenance of child. And the main area where DNA profiling and matching is used is related to paternity or parentage matters. As I said, it can be used for the paternity as well as maternity analysis. So, what are the paternity tests? These DNA paternity testing enables to determine whether a man could be the biological father of a child. We know that DNA is a genetic and hereditary material. All human beings, they inherit DNA from their biological parents. So, supposedly a child is there, we know that we have pair of 23 chromosomes. So, half of the DNA comes from the mother and half of the DNA comes from the father. So, a child will have combination of 50% DNA from the mother and 50% DNA from the father. So, DNA paternity test compares a child's DNA pattern with that of the alleged father to check for evidence for the inheritance and the most definitive proof of a biological relationship. The result of a DNA paternity test is either an exclusion that the alleged father is not the biological father, or an inclusion that the alleged father is considered the biological father. So, thus for a standard paternity test, we will say that 99.99% probability of paternity for inclusions or 100% certainty of exclusions. So, similarly, when the question arises for the maternity test also, the maternity DNA testing determines whether a woman could be the biological mother of a child or not. So, like a DNA paternity test, it compares a child's DNA pattern with that of the alleged mother to determine how likely it is that the child has inherited the DNA from the alleged mother. So, usually whenever these cases of the maternity test mostly arises, if a person is an adopted child, if there is a case of child swapping in the hospital, in those cases we have to go for the maternity test. So, the circumstances include the adoptee to prove biological relationship in the immigration case, to confirm that an embryo conceived through IVF was implanted into the correct mother, and to resolve the solution, as I said, where there are cases of baby mix up in the nursery. So, the maternity test, the child, alleged mother and biological father are tested, and the father's participation in the maternity test helps to exclude half of the child DNA, leaving the rest for comparison with the alleged mother. If the father is not available, then we have to go for the fatherless test, and which involves additional analysis, because whenever we have the DNA pattern of a child, then we assume that 50% which is coming from the mother and 50% which is coming from the father, then we match the other 50% with the mother. But in case if there is no DNA available from the father's side, then either we should take the sample from the father's siblings or the father's parents, where the match could be like around 25% or not exactly 50%, but maybe in the range of 25% to the 50%, which can still help us narrowing down that. So, with this DNA test, the genetic reconstruction can be done to know whether or not a child is related to the alleged father's close relatives. In this, we can go for the grandparents test and the siblings test also. Mostly they are used in the western countries, where the father is being married for the second time, third time, or the mother is being married to another person, and the children, they want to know who is their half siblings or who are their grandparents and who were their grandparents. In such cases, these tests are being confirmed. So, this genetic reconstruction, they requires complex analytical methods, and if only two of the father's close relatives are available to take the test, participation of the child's mother is required. So, in such cases, we go for the siblings test also. Mostly these tests, they came into the picture where we are talking about the common lineage, or if a person is asking the share in the property of their grandparents or from other their half brothers or half sisters. Usually, to get to know about the paternal relative, we go for the YSTR comparison test. What is YSTR? This short tandem repeats which are present on the Y chromosome are the YSTR test. So, as we know that there are 23 chromosomes, pair of 23 chromosomes in our body, out of which 22 are autosomals and 2 are sex chromosomes. These two sex chromosomes define whether you would be a boy or a girl. If the combination comes as XX, then you will be a girl, and if the combination comes in X or Y, then you will be a boy. But this Y chromosome, this only transfer from the grandfather to the father to the son. This Y chromosome is not present in the females. So, if we have to identify the YSTR patterns, then we can track the lineage up to the fathers, from the father to the grandfather to the great grandfather. But in that, we cannot go for the maternal relative or the female side. For the maternal relative, we have to go for the mitochondrial DNA analysis. This mitochondrial DNA, it is being transferred from the mother side only. Why the mitochondrial DNA does not come from the father side? Because the father side DNA comes from the sperms and they do not have the mitochondria through which the mitochondrial DNA is transferred from the father to the child. The mitochondrial DNA always comes from the mother side. So, to compare towards the paternal father side, we go for the YSTR analysis, and to know the paternal lineage test, which can confirm relationship to the long lost relatives and the paternal ancestors. So, why this test is most commonly used? It is used to provide the evidence in difficult paternity cases in which the alleged father is not available for testing. Indirectly, it can tell whether or not a child is related to alleged father's brothers and other male relatives who share a common paternal line. So, for the maternal lineage, we have the mitochondrial DNA test, which is used to support or disprove a direct maternal connection between two or more participants. So, mitochondrial DNA is present in males also. YSTRs are not present in females, but mitochondrial DNA is present in males, but it is coming directly from your mother or maternal side. For example, in a recent case of the ancient DNA analysis, where we have found the some excavations from the Goa of the queen relics, then we have to confirm whether they belong to the queen of Georgia or not. The DNA which we have extracted from the bones which were extracted from the Goa church. Then they were compared with the maternal lineage and we confirmed that they belong to the Queen of Georgia. So, these maternal DNA analysis can be determined and we can determine the child's relationship to a possibly maternal relative. So, in conclusion, I will say that to go for the paternity or maternity testing, we can use various platforms like STR analysis, YSTR analysis, mitochondrial DNA analysis, and with 100% surety either we can exclude or 99.99% surety we can include that this person, the biological father, mother, grandparents and siblings, they can be identified. Thank you.